Ladies and gentlemen, I invite you to take your seats as we get started with our second program. Understanding the interactions between cancer and the immune system can lead and is leading to some of the most concrete and exciting developments, particularly regarding targeted therapies. To lead a discussion to expand on this, please welcome Dr. Anna Barker, the Chief Strategy Officer at the Ellison Institute for Transformative Medicine of the University of Southern California. Dr. Barker served as Principal Deputy Director of the National Cancer Institute and Deputy Director for Strategic Scientific Initiatives, and Dr. Barker will introduce our distinguished program participants. Thank you.
Thank you, Jim. Good morning, everybody. Good morning. It's a great crowd. We're so glad you all came out to talk with us today, and so think about your questions as we get started. I actually am an immunologist by training, but I'm a polymath by choice. And so what does that mean? It means that all the things you heard last night, I really believe that, at least in oncology and probably all across biological science that it relates to disease, we really need to bring our friends from engineering and physics and mathematics in, because we're at that point, aren't we? That's where it all is going, and so we have to, the elephant in the room for biology is always we didn't like math, so have to be careful with that. So I wanted to set this panel up a little bit to tell you what we hope to do with you today. First of all, we have been studying the parts of cancer for a very long time. Now, what do I mean by that? It means that, and I had something to do with this, Dr. von Eschenbach and I were at the National Cancer Institute together until he chose to go to FDA and regulate everything that I did after that. So, you know, the NCI is a regulated entity, as you might know, so we do a lot of clinical trials there. But we started a lot of activities to set the stage for precision medicine when Andy and I were together, including working together with Francis Collins to set up the Cancer Genome Atlas, National Proteomics Program, the one you heard about last night, the National Nanotechnology Alliance, and the list goes on. But one of the things we did was to set up something called the Physical Sciences Oncology Centers to do just that, to bring all these disciplines together. And I think it's going to bear heavily on what we're going to talk about today. So immunotherapy, we could talk about a lot of things today, but we're going to talk mostly about immunotherapy because it's on everybody's mind. You're hearing a lot about it. You hear there are miracle cures, and we want to make sure that we get you excited about the right side of immunotherapy, and you need to know a lot about immunotherapy and understand it, I think, from top to bottom to be able to, what I'd like to make everybody in this room an advocate for immunotherapy. I think if we do our job well today, I think you'll see why. So this is a very old idea, goes back to Cooley in the 1890s, I think, when he made a toxin that was actually out of streptococcus in a benign organism, and he figured out that he had to actually heat up the streptococcus, and he made it into a toxin to treat cancer, and sure enough, he had some responses, and so that was the beginning of immunotherapy, although some would say it goes back to the Greeks. It's a very long in the tooth idea, but one that we believe has reached an inflection point. So when I said we started by studying the parts of cancer, that's what we've done. We're in either the genomics period or the post-genomics period, but I think we're in the middle of the beginning of the multi-omics period. So right now we're measuring patients for just about everything in terms of their omics, their genome, their transcriptome, their proteome, and their epigenome are all now possible to get on a chip. So that's become the basis for precision oncology. I will tell you that about 8% of people in this country who are eligible for precision oncology are getting that kind of treatment. So if you think we don't have problems in this country with who needs to be treated versus who's being treated, you're hearing a lot of conversation about that, and you're going to hear a lot more. So let me say one more thing about immunotherapy. Immunotherapy is not new, but the concept actually in terms of oncology is quite new. We're not treating a specific target when we treat the immune system. We're actually rebalancing or redirecting the immune system. So it's a systems approach. And I believe, as you heard last night, that cancer is a complex adaptive system. And if you put cancer in the same space as you have the immune system, now you have an extraordinarily complex system. And I would predict that in 10 years, that will be absolutely the only way we think about cancer. So everything that you know about what goes on normally in your body is still going on. Cancer doesn't invent anything new. It actually takes over processes that your normal system does every single day for you. But cancer, as you know, is a very wily creature and can take over those processes quite effectively. The last thing I'll say is I think of cancer as actually a disease of systems, but also a disease of dysregulated communication. So your cells at every level is having conversations all the time and making decisions all the time. And it goes without saying that once you dysregulate at the digital level, that's your genome, and that gets translated into changes in the analog information, that's the phenotype, that's you, you're going to have a lot of problems. And once that becomes metastatic, once that cancer evolves to the point where it can outwit all those wonderful defenses you have from your immune system, then we haven't done very well with metastatic disease in all the years we've been working on cancer. So we have a great panel today. I couldn't have chosen this panel any better because so if you look to my left here, you're going to see somebody who actually knows a lot about the basic science of immunotherapy and the immune system. And actually, I'm going to mispronounce Tom's name. I'm sure everybody does, but I'll do it too. So Tom Gajewski is the AbbVie Foundation Professor in Cancer Immunotherapy at the University of Chicago Comprehensive Cancer Center, one of our better cancer centers. Next to him is Carolyn Britten, who's the Vice President of Global Development and Early Development for all the programs at Amgen. And on the end over there is Carlos Becerra, who's the Medical Director of Cell Therapy and Cancer Research at the Hoag Family Cancer Institute, who has just been there 2 months, I think, right?
Yep.
Now, so I want you to know that this panel, if I go down this row of 3 people, I'm showing you all of cancer research today. So basic science and applied science, development and translation, clinical, and right next to patients all the time, changing their lives. And every one of these panelists is also a physician. So they're all patient-centric, and all the work that we're going to be talking about today is patient-centric. So Andy and I were talking before I came up here, and I said, you know, it's going to be interesting to think about immunotherapy relative to the last panel, because what we want to do actually here is to take this to a different level in terms of not just, you know, transfer of cells, some of them educated, some of them not, but how does that actually apply in cancer? What are the problems, what are the opportunities, and what do we do about it? So with that, I'm going to ask everybody to give me a couple of minutes on, here's the question. So this is a very old idea that's new again. So are you optimistic, and do you think that this might be the answer to cancer that we've all been looking for all these decades? 2 minutes. Just want to see what you think.
I guess since I'm first in line, I'll start. It's a real pleasure to be here. The point that you made towards the end, that cancer cells are not living alone, they're living at an interface with the host on multiple levels, including the host immune response, is really critical. It's been known for a long time that T cells of the immune system were capable of recognizing and destroying cancer cells. And in fact, in most patients, it was possible. I did a postdoctoral fellowship in Brussels with Terry Boone, one of the guys who discovered the first antigens that are expressed by cancers that can be recognized by the immune system. You could find T cells in the blood of a patient, mix them with the tumor cell line you derive from a patient, and kill the tumor cells. But somehow, in the patient, that didn't get lined up, right? So a big mystery was why. And what was found over the last 20 years or so is that in a major subset of patients, the immune system is engaged, the communication is initiated, the immune cells even come into the tumor. We call these hot tumors, right? T-cell inflamed tumors. But the T-cells are turned off again. And once the biology of that was figured out, these are called immune checkpoints. We now have drugs that are FDA approved in 20 different cancer entities where you block these checkpoints, you block the negative regulators, the immune system wakes up again and then destroys the tumor from within. These were fundamental discoveries that led to the Nobel Prize being given to Jim Ellison and Tsuku Hanjo in 2018. I'm friends with these guys, so I was at the Nobel party and ceremony and everything. There were some rock stars there too, and I played guitar in a band.
Jim was with us last year here. So.
Oh yeah, and so this was a transformational discovery. But it turns out there's a lot more to be learned. Oh, and by the way, we're talking about cure. This word came up several times last night. With patients with metastatic cancer, immunotherapy is what's actually curing a lot of patients. I treat melanoma patients. That's where a lot of this got started. And patients with metastatic disease can actually be cured with these immunotherapies. So that's the glass half full. I think that's the argument you wanted to hear from us. So I'll start there. We can come to the glass half empty where there's room to discover, I think, with a subsequent question.
Our first job here is to have you join us in really becoming enthusiastic, totally all in for immunotherapy. All right, Carolyn.
So I'm an oncologist. That means my default mode is optimism. And I am optimistic about immunotherapy. I came a little late to that realization. I started out in signal transduction. I'm another one of Dan Von Hoff's mentees, and had great mentorship from Dan Von Hoff, Gail Eckert, Dennis Slayman, mostly in signal transduction. I remember having arguments with Tony Rivas at Los Angeles and kind of at UCLA and kind of teasing him about immunotherapy. But, you know, you have to not only be optimistic, you also have to be adaptable. And we've seen some really great responses in immunotherapy, not only in hematologic malignancies, also solid tumors. Folks have been talking about co-opting the T-cell system, especially in hematologic malignancies, but we know that that also works in solid tumors. Solid tumors are behind hematologic malignancies, but just this year, we presented data with one of our compounds in small cell lung cancer. You've heard about re-engineering T cells. One of the things that we're doing is taking a T cell and linking it to the cancer cell through something we call a T-cell engager or a BiTE. And we've seen successes not only in hematologic malignancies, but as I mentioned, solid tumors, prostate cancer, as well as small cell lung cancer. So yes, optimistic.
Go ahead.
Short answer is I'm tremendously optimistic. I've seen it. I've seen the results of a lot of the work that has been going on for many decades by basic scientists in the clinic. I'm an early adopter because I do clinical trials with all these different type of approaches. Our previous panelist talked about that we still have a lot of work to do in solid tumors, that hasn't panned out yet. I have to say I disagree with that. I've seen it myself. You're going to see some data coming up, for example, in stomach cancer, refractory to everything else, having some wonderful results with modified T cells. Pancreas cancer, pancreas cancer, yes. We've seen results. We've seen very excellent responses with several different products. So we're going to get there. It's a matter of tweaking. Give us a few years and a lot of money, and we'll take care of it.
All right, so so part of what we're doing today, we have a lot of patients and patient advocates that are actually attending online. I know because I I sent this conference out to a lot of people that I know in the advocacy community, and it's, you know, I think it's really important that we bring this back and educate a little bit so everybody's on the same page. So, so Tom, could you say a little bit about what, you know, CAR T cells? Let's start with what CAR T means, and then how do you actually create those cells in a very brief description?
Yeah, so just to take a step back, the two main immunotherapeutic interventions that are well established now, I mentioned checkpoint blockade, where you release the brakes on the endogenous immune system. And you heard a little bit in the previous session about CAR T cells or engineered T cells, where you can take just generic T cells from the body and by genetic transduction put in a receptor that can recognize something present in that patient's cancer cell. So you're engineering it to be redirected towards the cancer type or sometimes the specific cancer in that individual. So two different approaches. Checkpoint blockade, you're reinvigorating the T cells that are already in the patient's tumor. That's maybe a little bit more straightforward. CAR T cells, you're from the peripheral blood, you collect T cells, put in the receptor of choice, grow them up in the laboratory, or now by companies. They ship that back to the center. The patient is conditioned to receive them. The product is infused. Sometimes additional therapies are given. And then the hope is that those T cells will recognize and destroy that patient's cancer. So these are two different categories of immunotherapies that are widely being applied and FDA approved in a number of different cancer contexts currently. But the technique is quite different, and what's been alluded to here is that the two fields are coming together to put them together in both hematologic malignancies and solid tumors.
Yeah, so Carolyn, talk a little bit about what are the challenges with actually making this work in terms of making CAR T cells sufficiently available in the quantities you need for clinical trials, let's say?
So I think probably there are others who can answer about CAR T cells more completely than I can. What we're working on are T-cell engagers, which are different from the CAR T cells in that they're off the shelf. The CAR T cells, I think Tom's already talked about the process, right? You have to harvest the cells from the patient, send them to the drug company, bring them back, engineer them, bring them back to the patient. Patient has to undergo a lymphodepletion therapy as well. For a T-cell engager, it's off the shelf, there's no lymphodepletion, and it's available immediately. And that's one of the advantages of the T-cell engager therapy. You can use a typical manufacturing platform. I shouldn't say that because it takes away from the magic, and it really is magic in making these things. Being able to produce these proteins is complex, but we've developed systems to do it.
So let's turn to how we actually administer these CAR T cells. Can you explain? You're seeing a lot of patients, right? And
Yeah.
So, is there anything special about CAR T cells versus infusing a chemotherapeutic drug? Let's say. I mean, so I'm trying to get to something here. What are the barriers to actually not just creating these cells, but administering them and actually? The last panel talked to some extent about the cost of these therapies. We're not going to go into that today, but suffice it to say they're very expensive.
There is a number of barriers. One of them is what I call physical barrier, which is a distance from where people live to where those treatments are being administered, and I think that was already alluded to in the previous panel. There's also a barrier as to who is potentially eligible. For example, just one example, in stomach cancer, which is a type of cancers that I manage,
With some of these modified T cells, only about 50% of the stomach cancers have the specific target, so that leaves 50% out. In addition to that, there's additional what we call exclusionary criteria that brings down the number of patients that can be effectively treated with this the smaller number, and the cost. So the cost, while the patients are on clinical trials, is paid for by the sponsor. However, you've heard about cost of Kymriah and all these commercially available modified T cells, which is we're talking about $500,000 plus plus additional cost.
It's a lot of money.
It's a lot of money. I think the technology will evolve in doing perhaps viral transfections will change once you move to, for example, from viral transfection to transposomes and different type of approaches. The cost will go down, and hopefully more patients will have access that way. And insurance will cover.
So we want to spend a little time on CAR-T because, we spent some time in the last panel talking about the promise of stem cell therapy, and all of these adaptive cells that we actually are creating now are, I think, very promising. I think it's very promising, but we have to all sort of integrate the fact that it's not easy to make them, it's not easy to scale them, and it's certainly not going to be easy to afford them. So let's turn back to why we gave out a Nobel Prize a few years ago for the checkpoint inhibitors. And the checkpoint inhibitors have probably become the go-to therapy for lots of diseases now. And certainly in combinations. And so if you go back in the history of CAR-T, it's long. We've been at this for a very long time. Equally so for the checkpoints. So Thomas, could you say something a bit about, what are checkpoint inhibitors? Why have they been so successful, or have they?
Yeah. So in terms of the years of research and development, a lot of basic science investment was required to identify these molecules on activated T cells that regulate them, that turn them back down. The immune system is sort of designed to rev up quickly in response to a pathogen usually, and then to turn itself off to avoid chronic inflammation and autoimmunity. So it's that balance that the system is built upon, and cancer is a chronic disease, and so you end up in this negative feedback phase of the immune response, and that's what the immune checkpoints are doing. The reason checkpoint blockade is so broadly applicable to a wide range of cancers, like I said, FDA approval in around 20 cancer entities, is because that same process of immune system going on and then turning back off is relevant for many solid tumors. And so you have a large fraction of patients with many different cancers who have, you might call it a smoldering immune response against their own tumor that's now held in check by these checkpoints. And so these drugs can be broadly applicable. The one that many of you are probably familiar with, the target is called PD-1, and the two main drugs against PD-1 are made by BMS and Merck. Just to be fair, BMS is nivolumab and Merck is pembrolizumab. Again, broadly applicable in a wide range of cancers. But how do we make that better? We have patients who have complete responses that are durable. We have patients more than 10 years out from the first checkpoint blockade trials who still haven't had recurrence of their disease. And in terms of cost versus benefit, I hope we can bring up the point of if a patient is cured of their metastatic cancer, they return to society. They go back to work, they go back to school, they're paying into the tax system again. And so cure means a lot in terms of sort of feeling relevant again, and then compensating for the cost of the therapy when you are returned to the workplace. But the non-responders is what the field is really focusing on. Most patients, more than half of patients, still don't respond, and we're figuring that out, right? One of the key things that I don't want to occupy too much time here, one of the key things that we found out is that patients who have a good immune response against their tumor versus a bad immune response, one of the drivers of that is actually their gut microbiome, the composition of the bacteria in the gut, which has been shown. We were the first group to find this in the context of checkpoint blockade, and it's been extended now to CAR T-cell therapy patients as well. There are good microbes and bad microbes, if you will, that occupy our gut, that push a favorable immune response against your own cancer versus not. And if you have a good microbiota, you're more likely to respond to checkpoint blockade. This is something we can change. We've licensed some of our own work to a biotech company to create special probiotics or bacteria that could be given in different forms. People are doing fecal microbial transplantation, where, believe it or not, you take a stool sample from a responder patient and you do a transfer to a non-responder patient. It can turn them into a responder. And then diet affects the microbiota. So there are other interventions that are being developed to try and push the favorable microbes. And that's just one example where, by studying patients who fail to respond, we're figuring out mechanisms why and then trying to change that so that we can turn them into responders.
So I've raised your level of optimism, and we've, I think, supported our optimism so far pretty well. But so when I said this is a complex adaptive system, the immune system, it really is, and it talks to every other system, and I hate the word talks. It communicates with every other system you have. And so when you look at the title of this session, it's Immunology and Inflammation. Well, your inflammatory response, as you found out in COVID-19, can be a good thing or a bad thing. I mean, right? I mean, the people that ended up in the ICUs with COVID-19, they had a wonderful immune response in their lungs, but the immune response overdid it. And so those people actually ended up being very ill. So your immune system, once that homeostatic set is out of sync, it's no longer balanced. It's moving toward really tuning up the immune system. That can cause toxicities that we know can be deadly. So let's talk about that a little bit in terms of, what are these major toxicities we could anticipate in patients? And if you think about an immune system that you're trying to actually activate and you overactivate, or you activate the wrong population of cells, what have you. So what are some of the toxicities we're seeing here? What do you say, Carolyn?
So if we talk about the immune checkpoint inhibitors, they can be associated with a series of events we call IRAEs, immune-related adverse events, where you basically can get inflammation in almost any organ system. You can get inflammation of your colon, have a colitis, inflammation of your lung, have a pneumonitis. The good news is that most of the time, when we're talking about these types of side effects associated with immune checkpoint blockade, it's manageable, very manageable. And the minority of patients would develop something that would be a real severe toxicity. The more common kind of IRAEs may be things that impact the endocrine system, which can be solved with, for example, replacing the hormones, things of that nature. When we talk about CAR T-cells and T-cell engagers, we're talking about a different kind of immune response, and that's called cytokine release syndrome. Basically, what happens is we activate the T-cells, and then we cause this cascade of proteins, for lack of a better word, but of cytokines that then can cause serious issues like low blood pressure, high heart rate, shortness of breath, and some of the things you talked about when people are seriously ill from a bad infection, this sort of inflammatory response. And the good news is that in the majority of cases, serious CRS the chances of that happening is very low, and we know a lot about how to prevent it and how to treat it. So what we commonly do now is we give steroids, which kind of blunt the release of these cytokines. And we also, once the cytokines are released, we can block the effect of the cytokines with a drug called tocilizumab, for example, which is an IL-6 receptor blocker. But we're doing some really cool things to re-engineer our drugs to try to get away from CRS altogether. And some of the things that we're working on include what we call dual bites or and gated bites, in which the molecule is not only binding to one tumor-associated antigen, but it's binding to two tumor-associated antigens and the T-cell. Now, why is that important? There are a minority of tumor-associated antigens that are specifically expressed by cancer cells. So some of these tumor-associated antigens could be expressed by normal cells. So if you're targeting with two tumor-associated antigens, then you're really being more specific to the tumor. So that's one approach. Another approach is...
That's a breakthrough, isn't it? I mean, that
it's super cool.
bispecific antibodies are a breakthrough.
And another approach is, well, that would be a trispecific. So a bispecific would be the T-cell engagers that are already approved that bind one tumor-associated antigen to the T-cell. A trispecific binds two tumor-associated antigens and the T-cell. So that's one approach. Another approach would be what we call masking, or somehow hiding the bite or the T-cell engager until you get to the tumor. And then once you're in the tumor microenvironment, you cleave or unmask the protein, and then it can do its job.
So isn't it interesting, Carolyn, really what you've been talking about is taking a complex system and redirecting parts of it, blocking parts of it, enhancing parts of it. Very interesting. It's an engineering. You're really a molecular engineer at this point. So are you seeing anything, Carlos, that is unexpected in patients that that really surprised you, that you thought, geez, I would never have thought that would have happened other than the miracle cures? I mean, are you seeing toxicities that you think that we can't manage or should worry more about?
No. One of the things that I found out over the years in dealing with these living drugs, for example, the CAR-Ts, I call living drugs, is that they're fairly predictable. So you give them, generally speaking, you give them the infusion of the living drug to the patient on Monday, and on Wednesday at around 10 p.m., I'll get a call.
Could you speak more into your microphone?
On Wednesday around 10 p.m., I'll get a call from the floor nurse. Oh, the patient is starting to having this and that and so on and so forth, and we have a protocol for that. So it's just a way of the cells reacting to and engaging with their target, and as Carolyn was alluding to, we already have medications to try to quell down the problem and take care of it. Therefore, the risk of severe toxicity is much, much less.
So, Carlos, when you're sitting in front of a patient and you have a portfolio of ideas, say a GI tumor, you have a portfolio of ideas that standard of care for certain kinds of cancers. How do you make a decision that an immunotherapy would be better than, say, a targeted therapy, a small molecule?
So there is a specific blocks, let me put it that way, in the clinical trial where we don't go rogue and you say, for example, somebody who has never been treated and go say, well, let's offer this to this patient yet, as long as there's some standard therapy available. But beyond that, once the patient fits a certain pattern, we discuss it with the patient and the family it's critically important to discuss it with the family too, because they're going to be supporting the patient. Once you explain it, if they're agreeing to it, we proceed.
So, for everybody on the panel, you know, we're at that inflection point where immunotherapy is starting to be certainly not routine yet. I mean, I think it's still for some tumors, I mean, and for certain checkpoint inhibitors, I think, well, at least one checkpoint inhibitor we all know about. We seem to treat most cancers with one of these checkpoint inhibitors. There are 20 plus checkpoint inhibitors, by the way, approved by the FDA. The person who oversees oncology at the FDA often asks, how many checkpoint inhibitors do we really need, since they're targeting the same sort of antigens. But the question is, it is still a complex system after all. And you have all of these things available. So will we see a day when immunotherapy becomes monotherapy, or is it always going to be combinations? What do you think?
There are several different flavors to that question. First of all, in terms of being standard of care, I just want to maybe push back on you a little bit there, because in metastatic melanoma, for example,
yes.
Even if somebody has, if someone has a mutation in a particular gene, it's called BRAF. We have targeted therapies available and immunotherapy. The feeling was that if patients got immunotherapy first before targeted therapy, they did better, and now that's been proven in a randomized clinical trial. So immunotherapy becomes first. Now that's for metastatic disease. Then immunotherapy, checkpoint blockade, was moved to the post-surgical adjuvant setting after somebody had surgery and had high chance of recurrence. Immunotherapy now is the standard there. And now, in the past couple of years, it's been moved to what we call the neoadjuvant setting. So somebody presents with a large tumor, lymph node mass. Normally they would get surgery first. Now we get immunotherapy first, and that's been proven in a randomized trial to be better than doing surgery first.
Also in breast cancer.
Yeah, and with immunotherapy though.
Yes.
In melanoma. So now most patients would get immunotherapy before their surgery even. So it's being pushed earlier and earlier in the treatment scheme. And I just want to argue it makes logical sense. Once the cancer divides and spreads, there are more mutations that accumulate. You're more likely to get tumor cells that become resistant to the therapies that you're considering. This gets back to sort of tumor evolution that was referred to last night in the mathematical session. It makes sense to give the therapy earlier to improve the possibility of cure. Then other checkpoints. This has to do with biology. So we know that there are patients who don't respond to the current checkpoints, even though they've got a rip-roaring immune response in their tumor. By studying those T cells, we've discovered other negative regulators, and some of those are being targeted in biotech startups that I'm part of as well, right? So in terms of too many checkpoints, I think we just have to let the biology drive what we're doing and make new discoveries and see what the next level immune regulators are, and so on. So biology drives.
So we've had quite a journey in immunotherapy, especially in the last few years. As you heard, melanoma was probably the first tumor beyond the hematologic malignancies to really benefit from this. And pediatric cancer patients now have been and are being treated with immunotherapy. So it's becoming quite acceptable across, I think, all types of patients. So let's talk about another problem, though, that is close to my heart, and that is how you find those patients. And so one of the issues we have is we would all like to know that we're going to go in and get access to this kind of therapy. We all have an immune system, and we aren't talking about this, but maybe we should. You should never get cancer, right? If your immune system is on target, why do we get cancer in the first place? That's a really good question, and the answer is you probably do, maybe several times in your life, for all you know. And we now have a whole group of assays coming out that say, and I think it's going to be an inflection point in cancer research, that they can find your cancer at very early stage. Now, I can't tell you that we know anything what to do, what we're going to do about it in, say, stage 1, but let's say it's stage 3. You could downstage that cancer, but more likely your immune system might just take care of that. So, we're going to be in an interesting, challenging period here, I think, for, what do you do with these assays? But let me come back to that, and let's talk a little bit about the biomarkers for this whole field. Kind of where are we? And, a small percentage of patients react, but we don't know how to find them very well yet. And so we've got biomarkers like tumor mutational burden. In other words, what's the status of mutations in a particular patient? It's not a very good measure of very much, but we also have these antigens, the checkpoint inhibitors. We can do pretty well with that, I think, Tom. So where do you guys think this is going in terms of how do we identify patients who are going to respond, and as importantly, the ones that are not going to respond? Carolyn's picking up her microphone. She has the answer.
I thought it was my turn. I feel like this is a tough question. A lot of what we're talking about in terms of CAR T cells or T-cell engagers are specifically designed to work against cancers with specific tumor-associated antigens. So there, you either pick a tumor that has a high chance of expressing that tumor-associated antigen, or you can actually test the tumor for expression of the tumor-associated antigen. But not all patients that that express tumor-associated antigens are necessarily going to respond to the agent, just like any other anti-cancer therapy. Response is not guaranteed. And so this is very, very tricky. And one of the things we need to do is look at the patients who respond and who do not respond and do tumor biopsies so that we can see how do the patients differ? How do they differ at baseline, and how do they differ after treatment? And frankly, that's asking a lot of the patients who go on these clinical trials. That's asking them to undergo biopsies that are not done for their benefit, but are really done to kind of further science. And that's a huge ask. But people who have cancer in general want to see advances, and if not for themselves, then for others. And in the 20 years I was doing phase 1 clinical trials, I was often humbled by what patients were willing to do to help others.
Coming back to this concept of cancer being a complex adaptive system, and actually it's a system that's working all the time, even in the presence of cancer, maybe not as well. But what about stepping back and thinking about, what about assessing the state of the immune system in the patient as a way to come closer to identifying those patients that are going to respond? What do you think, Tom?
Yeah, so this is right where the translational research is at the moment. Usually, people used to think that metastatic cancer patients were globally immune suppressed and there was no way an immunotherapy could work. That might occur, but with solid tumor patients, it's a very small subset who get to the point of being sort of cachectic, losing weight, and being globally immune suppressed. Most patients have a still responsive immune system. We know that because we measured immune responses to COVID vaccines in all of our patients, and they made fine immune responses, right? And flu vaccines, et cetera. But the key is, do you have immune cells specific for the tumor, and where are they, and what are they doing? What's their activation status and their functional status at that moment in time? And what's making them work, and what's making them not work? And all of that can be measured with very advanced omics techniques now. And most of the action is at the level of the tumor microenvironment, so tumor biopsies are required. And you've heard about RNA-seq, or looking at gene expression in cancers. That includes gene expression of immune cells. There you can do sequencing of the mutated genes in the cancer, and some of those are connected to immune response good or bad, working or not working. And now, in the past several years, there's been a lot of work doing what's called single-cell RNA-seq. So you take a tumor biopsy, you chop it up into all the cells, and you sequence the genes in each individual cell, and you put it together like a confetti, and you see what genes are expressed by all the different cells in the tumor cells, the blood vessel cells, the immune cells. And now, this year, there's been a huge application of a new approach, which is called spatial transcriptomics, where we can take a biopsy, and in a section, we can look at all the little dots of all the different cells in the tumor and just leave them in place geographically and see who's making what. And we just have a paper that's going in where we found out something really important in responders to immunotherapy. There are two cells talking to each other at a moment in time, and if those two cells are talking to each other, those are the responders. So it means we've got to bring new cells in that have to communicate with the T cells in order to expand efficacy. So a lot of technologies are going into this, and it's the dynamic interplay between the tumor cells and the host cells that's the key to success.
I think it's really important. Carlos, you look like you're sort of disagreeing down there.
No, I follow, of
You're
course,
thinking.
the literature, and I can tell you one thing is that the cancer cells are very clever. They find a way to co-opt or bring in not just the immune cells, but other cells into the equation to their favor. And you mentioned about the fact that signaling, that there was a problem with signaling between the cells and so on and so forth, and that's for real. You know, the investigators have found that going back to, for example, endothelial cells, and how the cancer cells take those cells and called the endothelial cells and use them to their advantage, and take other cells and use them to their advantage, and so on and so forth. So it's quite complex, but if we all work together,
Yes.
it can be solved.
So, Carolyn, some patients are actually resistant to immunotherapies right out of the gate, right?
Patients who are, all patients that I had wanted immunotherapy, but you're saying the tumor was resistant out of the gate.
That's a better characterization. That tumor is very resistant. That patient's all over it, right?
I had a lot of patients when I was in practice who wanted the Jimmy Carter drug.
I think Carolyn has a future as a stand-up comic.
So when we talk about, there are a lot of cold tumors, right? So in practice, I dealt with pancreas cancer. That was one of the areas that I concentrated in. That is a notoriously immunologically cold tumor.
You want to say what that means, cold, hot?
Hot would be something that is immune responsive. Cold is something that is immune resistant. A cold tumor might have a thicker stroma, or an environment that is not permissive for T cells to enter, for example. So yes, there are some tumors that start out resistant, and we're doing, I think the field in general is working on ways to try to make cold tumors hot, and that's, has to do with turning on and off various checkpoints, or manipulating the microenvironment and different things like that.
Or were you even using viruses to make him hot, or using bacteria to make him hot? Back to Cooley's toxin from 1893. So yes, it's happening. There's a lot of different smaller companies that are in labs that are working on this.
Yeah, I agree. It's really so I have to say, just for the audience's sake, a lot of things that we don't understand, we just make up words for it. So you know that, right? So we talk about exhausted T cells. You have the feeling that these T cells are just kind of laying on lounge chairs or something. So it's kind of a strange term. But eventually, you find out that there are T cells that, for a whole bunch of good biological reasons, are not able to respond. And so, Tom, can you say more about that? Because is that I mean, is that you just, the patient would never respond, or is Carlos right that maybe you could turn that tumor on?
This is,
Or off?
it's the latter. This is a sort of a different terminology for the same process we've been describing using different words throughout the morning here, that when you do have, let's say, a hot tumor, you have activated immune cells that have come in to try to destroy the cancer, but they're shut down in a way. Those T cells are what have been termed exhausted T cells. I hate that term. It's actually not precise enough, right, for some of us. It seems like they're just pooped out and they just need a good night's sleep, and the next day they'll come back, but they're turned off by specific molecular mechanisms, and by reversing that, you can get them to do their job again. And that's in part what the immune checkpoints are doing. And I just want to come back to this really key point that was raised, which is the cold tumors, right? The non-T cell inflamed tumors, they don't have any T cells in their tumor very often, exhausted or rested, right?
Yeah.
A lot of people in the field, including our own group at University of Chicago, we're figuring out how to get those T cells in. It's a complex system. It's not one factor that's missing. It's a cascade of events missing in those tumors to try and re-engage or initially engage the host immune response to get those patients on the same level playing field as the hot tumor patients, so we can get these immunotherapies to work.
Before we run out of time, I want to make sure if we have questions here that you want to have a chance to ask a question. We will, if you know if your tumors are hot or cold, that would be good to start with.
I heard this morning that the studies as well as the treatments are very, very expensive. So my question is there an upper age limit to the studies or the treatments?
So for the approved checkpoint therapies, there's no upper age limit. And in the beginning, because of the toxicity risks, there can be side effects, and if they do happen, they can be they have to be managed quickly, but you worry in an older individual. I have to say that some of our best complete responders in melanoma have been 90-year-old retired people. I'm in Chicago. These, depending on whether they grew up as Cubs fans or Sox fans, they're retiring in Arizona or Florida, and then they fly up for their treatment, they get their dose, and then they go back to their retirement home or condo, their winter home, right? And they tolerate it just as well as younger people, and the efficacy is very similar.
Carolyn?
Even with some of the more toxic approaches, such as CAR T, when I got involved early with some companies in designing the study, we really didn't put an age limit. I did it on purpose. And I've treated patients up to age 83, as long as they're fit, with this approach.
Interesting, too. I mean, we didn't really talk about this specifically, but for patients that are cured, essentially, of their cancer, you have memory cells. I mean, so it's a very interesting transformation that occurs in these patients. So did we have another one?
Hi, my name is Dhruv Roy. I'm from Heliogenomics. I'm glad that Anna mentioned about biomarkers for early detection. And question for the panel, how do you see this precision medicine is evolving driven by genomic and multi-omics data?
Anybody want to take that?
Regarding immuno-oncology, I'm assuming, right? That's your question? Well, if it becomes once we validate it as a predictive or prognostic marker, as you can realize, it will be very useful in that setting. Early on, I think you and I talked a little bit about this early on. If we can look at the general population and find out who's at the verge of having a cancer that's going to break through, that might be another very important setting. As long as we know what to do with that information,
yes.
And if we were able to change the natural course.
So we're going to run out of time, but I want to give our panelists a minute each. This is a real challenge to tell us what is the most important thing we can do to move this field forward. And I say that because if I were running the world, I would be putting a lot more resources into this field, and we're not. And I don't know, I sit on the National Cancer Advisory Board in Washington, but I don't see it. I mean, we have a moonshot. I think we need a Mars shot for immunotherapy. That's my own personal bias, so I'll just put that out there. And if I were still in government, we'd be having, you know, one of these, not a human genome project, maybe an immunotherapy national project, but I think it really needs a shot in the arm. So start with Tom. 1 minute. What would you do?
Yeah. For me, it's understanding failure. So now we have drugs that are effective in a large fraction of patients, and we need to figure out why they're not working in the other fraction. And so we need to, and this is what we do in our program, we're banking everything from multidimensional omics, germline DNA, serum metabolites, tumor exome sequencing, tumor RNA-seq, gut microbiome sequencing, et cetera, and understanding the multidimensionality of mechanisms of resistance. So the patient now becomes the center point, and we call it reverse translational research. We figure out all the different dimensions of mechanisms of failure, like all the different ways your car can break down. There's one way to put all the parts together to make your car run. You take any one part away and it can fail. So too with immunotherapy. We're cataloging all of those. It's an AI problem in many respects. And each of those patient observations goes back into the lab for a clinically relevant basic science project and a discovery platform.
Well, Tom, just like we're finding, you know, we're finding drugs that work initially in one cancer, and then we start looking at other cancers, and obviously the pathways are very overlapping. It's a complex system, and I think, you know, I think there is an opportunity here to treat every patient, maybe in combination, but I think there's an opportunity to treat every patient with immunotherapy in some ways. So, Carolyn, what do you think?
For me, it's really finding ways to work together. I mean, we all spent 3 years, maybe 2. It seemed like 5 in our own little silos, in our, you know, on our WebExes. But we've really got to figure out ways that we can partner with academic institutions and industry and remove some of the gunk that gets in the way. And by gunk, I mean bureaucracy. And we need to find ways to partner with patients, especially, and take into consideration what are the things that are most important to them, and what do they consider as reasonable to undergo on a clinical trial, and what is not reasonable. And then I would totally agree with what Tom said, and learning from past experience and failing forward.
Carlos?
I advocate for the community here. The majority of patients are managed in the community. And again, this requires a partnership with industry, basic of labs or basic science labs. But we all have to understand that the human being is very heterogeneous, and it requires a lot of information, a lot of input from a lot of patients and different backgrounds in order to answer this question. And that information will come from patients that are managed in the community.
So put the patient at the center. Put the patient at the center and start really developing an understanding of, you know, what is the state of that patient immunologically from the time they become a patient until they're treated, and then post-treatment would be very helpful. The other issue that was raised I thought was really quite profound, and that is, in many ways, this disease is the disease of outliers. We should study the outliers a great deal more, the super responders and the non-responders, and I think we could use that database. I hope that what you take away today is that this session actually is talking about immunotherapy, but we're really talking about a change in the way we view biology and the way we view the disease we call cancer. Essentially, it's really a disease of a very complicated, complex system. And people always ask me, why are we not making progress a lot faster? Well, we have been studying the parts of cancer, and that was important. Very important to know the changes in the genome, very important to understand all that. But now we're at a point now we need to understand that from the point of the patient and the system that that patient represents, and I should say the complex adaptive system that that patient represents. At our institute, we talk a lot about coarse-grained biomarkers. That is, biomarkers that are more reflective of the state of the patient, and as somebody that works at the molecular level, we spend a lot of time down at the molecular level. How does that translate into what you see in patients, especially around the immune response? But I think we're all optimistic, and I think this has been a great discussion. I want to thank our panel and thank our organizers, Dr. von Ischenbach and Marlene Malik and Ellen Siegel, who can't be here, and all of you for being here, and all the patients for helping us in science to understand that it is the phenotype, it is the patient that's important, and we're very aware of that, and we need more of those kinds of partnerships. Last thing I'll say is everyone up here has also a relationship to the private sector. They've started companies, or they're in a company, or they're part of that process. We need more of that in terms of translating this information much more rapidly into patients. So thank you all for listening today.
Ladies and gentlemen, that concludes the second panel of the Nixon National Cancer Conference. We ask that everybody exits the room to allow our catering team to come in and place lunch. We'll join back here at 12 o'clock p.m.